Well, hello. I am with Donny Budinsky from Standing for Truth Ministries. Um, could you give a brief introduction of yourself, Donny, before we begin?
Yes, my name's Donny. I'm the founder of Standing for Truth Ministries. Been running the ministry now for about 5 years, doing it full time for sometime between 2 and 3 years. And so we host a lot of debates, interviews, conferences, lectures, presentations. And so for those who love that kind of content, make sure to check us out. I've actually done a show with you, as you know, maybe about a month ago on Chromosome 2. And so, Brian, you're an excellent host, and I appreciate you having me on again, brother. I'm really, really excited for this.
No problem. Yes, we did a show about a month ago on Chromosome 2, and now we're back now doing a segment that's going to be part of a larger video, response video to Erika from Gutsick Gibbon. Incidentally, what do you think of Dr. Carter's video that he produced responding to Erika? I thought he did a really good job, and he was spot on what he said about Josiah Siemens' paper.
Absolutely. Dr. Rob Carter, I mean, he's one of my favorite young earth creation scientists. He's been a huge blessing and just a huge influence on my journey as a young earth creationist. And so I thought his video was very good, as a lot of critics were under the assumption that Richard Buggs had completely changed his mind, basically from the 84% number up to the 96% number.
Yeah,
But as you know,
I wonder how he got that, Erika.
Right.
Why they got that idea from.
Well, and Richard Buggs confesses that geneticists don't typically compare whole genomes. There's no real agreement on how to even do such a thing. And so large unalignable regions are completely left out of the equation. Copy number variations, gaps, size differences, it's left out, and they're basically comparing these alignable regions. And so sure, you get 96%, but by definition, once you start bringing in other portions of the genome that weren't considered, the number is going to be a lot lower than that. That's logical.
Yep. Yes. And Donny is basing that on the email that I had received from Dr. Richard Buggs himself.
That's right.
In which case I forwarded it to Donny. Hopefully by the time everybody sees this video, they've already seen that email. So y'all know what you were talking about here.
And he agrees, correct me if I'm wrong, Brian. Richard Buggs agrees that all differences should be considered.
Yes, that was he said that's his view. He thinks that all differences ought to be considered, not just some differences. So that's huge. Yes.
Basically, if you were to include all of the unalignable parts, such as large duplications or large indels, insertions, deletions, the genomes would be even more different, probably around 80%. And so these analyses are actually very generous with the similarity. And we're basically looking at a difference minimum rather than a maximum.
You heard
that should be pretty basic. I mean, if Richard Buggs and Josiah are admitting that the genome comparison between chimpanzees and humans is only 96%, but much of the genome has been left out of the equation, obviously the more of the genome that you do bring into the equation, the more that number drops.
That's correct.
We're going to get into all that.
It's been a little bit since I rewatched that clip. So, but I know you probably taking some detailed notes, and we're going to get into that. So let's, without further ado, let's share my screen.
And ask some questions, because I don't think I heard anything on chromosome 2 fusion in your presentation. Could you explain that real quick before we jump on that stuff?
Oh my God.
Yeah, I actually have done entire presentations on that can get very technical. So in the so-called chromosome 2 fusion, evolutionists believe that 2 chromosomes fused from the 8 genome to form human chromosome number 2. The reason they had to go that route was because humans have a chromosome number, a diploid number, 46 chromosomes, and 48 chromosomes. And so they did find some gross similarities doing cytogenetic spreads.
We're going to pull up human chromosome 2 fusion. I think Ken Miller has a really awesome diagram. So we're just going to do that. I'm going to show it to you. I'm going to talk about it. Okay, present, share screen, initiating screen share, going over to human chromosome 2 fusion. Okay, all right, so human chromosome 2. You guys, back in the day when we first did karyotypes for the great apes, it was something of a surprise, I think, that humans had 46 chromosomes and the great apes had 48. The other great apes had 48. So a prediction was made in like 1972 by Turlow and Grouchy, where they were like, okay, well, like what if two things happen, right? Either all of the other great apes, like the ancestral condition was human-like, and it had 46 chromosomes, some Miocene ape had 46. And then in each grade ape group, they subsequently experience some kind of fission event to give them 48. Or alternatively, the opposite, more parsimonious answer, which is that 48 chromosomes is the ancestral condition and modern humans experience some kind of fusion event. So with this prediction set, they were like, a fusion event probably occurred, and based on like kind of brute banding, they were like, it was probably a human chromosome 2 for two reasons. One, because human chromosome 2 is big, and two, because human chromosome 2 has remarkable syntony or similarity with chimpanzee chromosome 2A and 2B, which at the time were known as 12 and 13. So what you're looking at here is sort of the hypothetical situation here. We have our ancestral chromosome here and our other ancestral chromosome here. And if the fusion occurred, there's going to be some like pretty classic calling cards to let us know, like a smoking gun, if we're looking at a fusion site. The first thing is, we should find a fusion site. It should be in the middle of the chromosome. So like, how do you find a fusion site in a chromosome? The answer is actually really easy, because the ends of chromosomes are capped with what are called telomeres. Telomeres are highly repetitive sequences of DNA, and they protect the chromosome during things like division from fusion, basically. And they also protect the genetic material that's kind of further upstream from these telomeres. So you should find a fusion site by finding telomeres in the middle of the chromosome at the fusion site. And these telomeres are clockable on the basis of a highly repetitive sort of telomeric motif.
Yeah, right now there's not much to refute. I mean, she's just going over essentially what I went over for the first several minutes of my presentation I did with you, Brian. Just the basics, right? How they discovered the so-called fusion site using karyotyping, right? During that method, you can essentially isolate cells as they're dividing during mitosis. And so that gives a visualizing of the chromosomes, and it generates these banding patterns. And so they can look at similarities in these banding patterms. And so that's...
Well, I will add one thing. Telomere fusions don't normally occur in nature. Very rare. That cancer patients are only like an example of a telomere fusions, but everything's kind of going crazy in the cells and that sort of thing. So as far as I can tell, we don't really normally observe them. Some people claim that they happen in the past in animals, and some peer-reviewed papers of pigs and things like that have alleged chromosome fusion, telomere fusions happening, telomere fusions happening, but it seems
they infer
unusual.
Right. There's a couple inferred examples, one in pig species and another one in equids.
Yeah.
And so those are inferred. Those weren't necessarily observed to happen, but they're looking at the genomes of both organisms, and based on the characteristics seen, they're inferring, okay, this was a telomere to telomere fusion.
Glenn Williamson from RUHIP sent me a number of emails, and among them had alleged fusion event in ants. Do you know anything about that?
An alleged fusion in ants.
Yeah.
He did send me that specific one as well. So I think that's another example of what they say is a telomere to telomere fusion. But I did a lot of study on her pig example and how she essentially compared it to the human chromosome 2 site. But what's interesting, and I had some back and forth with RUHIF over this issue, and the so-called pig site, I believe it's in this pig chromosome 6, the area is thousands of base pairs long. I think it's between 40 and 60,000 bases. I could pull up my notes for that specific issue. Point is, it's way larger than the 798 base pair region in the purported fusion site. 798 base pairs versus 40 to 60,000 base pairs. In the chromosome 2 spot, there's only, roughly 10 or so, more or less, intact TTA GGG sites. And then there's about 42 intact CCC TAA sites that are unbroken, right? That are actually in a tandem repeat. In the so-called pig chromosome 6 site, there's about somewhere between 1,000 to 2,000 examples of TTA GGG. Well, that's a huge difference from about 10 in the so-called fusion site. And then for the CCC TAA in the pig chromosome 6, there's about 900, somewhere between 8 and 900. That's a huge difference. Yeah, that sounds like the remnants of a fusion, but it's not apples to apples is my point. I think you're getting, in the purported fusion site, there's only about 10 instances of TTA GGG and about 42 of CCC TAA, and the entire site is only 798 base pairs. It's less than 1,000. The so-called pig site is way larger, and it has way more of these instances of TTA GGG and CCC TAA. So where she wants to make that comparison, it's not an apples to apples comparison. If the so-called fusion site, Brian, if that site was 60,000 base pairs with somewhere between 1,000 and 2,000 instances of TTA GGG, then they'd have a good case. Yeah, that sounds like the remnants of a fusion. That's a large signature, but we don't find that. We don't find in the so-called chromosome 2 site what we find in the pig chromosome 6.
Right. And I know that Ruhip also sent me a link to laboratory experiments that have been done on telomere fusions, but I was trying to explain to him, okay, my argument, and as far as I know, all arguments on this is including yours, is really probabilistic. We're saying that we're not saying that it's impossible for telomere fusions to happen, but rather that it's improbable because of the rarity of it happening or occurring. And the only reason you would cite a laboratory experiment is if I was arguing that it's physically impossible for telomeres to fuse, and that's not my argument. And so I think he like when we cite laboratory experiments on radiometric dating, radiometric decay rates not being constant, the other side actually are saying that it's physically impossible for accelerated nuclear decay to happen. That's why we cite the laboratory stuff, because it shows that it's not impossible. But we're not arguing that it's impossible for telomere fusions to happen, only that it's rare. Correct?
Right. So the site in the pig chromosome 6 is about 38,000 base pairs. And there's a lot of, also, if you look at the site itself, there's a lot of ends. What that is it's meaningless data. They don't really know what the sequence is. So it's not even
Rob Carter
fully,
actually talked about that too in the video.
Right.
The ends being like areas that they could not identified. And I noticed that too, like when I was looking at blasts and stuff like that, going at the different sequences, I noticed there was a lot of ends. And when I was looking up how to do all this with blast, I found out that you can't even put the ends in the boxes to make comparisons because it's nonsensical. I mean, they don't know what that information is, essentially. Correct.
Well, from my understanding, the pig genome hasn't been updated since 2017. And so we don't have the kind of...
A lot of these, from what I understand, they don't have the money and funding to produce a lot of these genomes. The human, because of all the illnesses and diseases and things like that, they put a lot of money and time into trying to sequence the whole human genome. But when it comes to like other organisms, even the chimpanzee one, it was done with very limited funding, very limited time, very limited staff. And so they end up just kind of putting something together. They did a lot of shotgun sequencing, and it a whole lot of time and effort wasn't really put into it. And I was just curious about other organisms as well. Do they also have a similar problem where not as much time and effort and money and resources are being put into sequencing the whole genomes of other animals?
Right. Yeah, with the Human Genome Project, I mean, billions of dollars has been put into studying the genetics of humans. But when it comes to other creatures, not as much time, energy, and effort has been put in. So we don't have the sophisticated data for something like pigs that we'd have for humans. But what we do know about the so-called chromosome 6 fusion within the specific pig species that Erika is pointing to, the entire site, again, is almost 40,000 bases. There's somewhere between 1,000 and 2,000 instances of TTAGGG. Well, how many instances do we find in the so-called chromosome 2? Again, we only find, about 10. That's a huge difference. So put it this way, there's a big signature in the chromosome 6 within this pig species. Brian, you and I, we don't deny that chromosome fusions happen. Chromosome fusions are a manifestation of genetic entropy. Okay, genomes going downhill. These are unilateral mistakes. And so we understand that they happen. The question is, did a fusion event happen in the human line? And the signature that we find in human chromosome 2 is very small compared to something like the fusion that we find in this specific pig species genome. Yeah, 38,000 base pairs with a lot of instances of TTA, GGG, and CCC-TAA. Yeah, that's a big signature. That's probably the result of a fusion. That's what we'd expect to find. But what we expect to find if humans experience a fusion, we don't necessarily find that.
What'd she say about, I know we dealt with this in our original video, but we might as well respond to that position because she brought it up, about the telomeric repeats. And I guess she thinks, well, it's got to be a telomere fusion because you got the same repeats being found here. Now, interesting enough, when we responded to some comments she made against you, she made the statement that that it needed to be in high density, and that's part of the argument. Except, like you said, she doesn't say that every single time she said it, and she certainly didn't say it here. She said rather because it had those telomeric those repeats, that this must be a telomere fusion. And as you pointed out, that this is actually found in other places. Is that correct?
Right. Good question. So Chris Roop and I, we specifically found other instances of the TTA-GGG, right? Your telomeric sequence in forward, and then your reverse complement CCC-TAA. We found many instances of those sequences in a head-to-head organization. This mirror image pattern, essentially. We found many examples of that pattern all throughout the genome, but we found a fairly large signature in chromosome 9, where you have TTA-GGG, TTA-GGG, one after another, unbroken. It's actually more pristine than the so-called fusion site. And then you find the CCC-TAA connected. So we find this head-to-head organization in chromosome 9, but nobody would say chromosome 9 is the result of an ancient fusion. And so these sequences exist, these internal telomeric sequences exist all throughout the genome in varying densities, and they're there for functional reasons involved in gene expression, chromosomal stability. Erika wants to look to the high density nature of the so-called fusion site, because you're looking at something like a 798 base pairs versus, I think, the chromosome 9 site. I wrote an article and then I presented it with you about a month ago. So in a nutshell, without pulling up my article on it, it's about somewhere between 40 and 50 base pairs. Now, when she says, but the chromosome 2 area is high in density of these repeats, that's true. But to then extrapolate that to mean this is the ancient remnants of a fusion, that is logically fallacious, because high dense, it's high in density compared to what? This is relative, it's arbitrary, because she wants to say, well, because it's a fusion event. No, because now if you invoke a fusion event, it's not high in density because it should be higher. There should be more sequences, like we see in the pig, chromosome 6, 38,000 base pairs, right? So it's kind of like when the critics say, this universe looks old, Brian. Oh, really? Can you show me another universe? Universe 2, 3, 4 to compare this one to?
That's actually the reification fallacy. When you say something looks old, the reason when we say someone looks old, like when I say Donny, you look young, I'm comparing you to other people that look old. So I'm actually using that in a very loose or some figurative manner. But when someone says the universe looks old, you'd have to have another universe to compare it to, and you don't have that. And so you're just simply assuming that it looks old because you're assuming you're the older position or something. If you claim it looks old, because it doesn't look. I mean, what the man on the moon has wrinkles on him. Oh,
Right. So right here, me and Chris Roop, we discovered, I want to zoom in. We discovered a similar signature in chromosome 9. The telomere-like hexamer sequence and its reverse complement, that's the CCCTAA, are arranged head to head. So basically, if they want to say the chromosome 2 spot is the result of fusion because we have a head-to-head arrangement of these sequences, well, wait a minute. What about the other examples of that throughout the genome? Those aren't the result of a fusion. So why would the chromosome 2 spot be the result of a fusion? It sounds like it was cherry-picked because of its high density nature. So right here is the chromosome 9 site that we discovered. So notice here's the sequences before and after. And in red and blue, we have the head-to-head arrangement. TAGGG, TTA, TTA. So you can see how they're all connected, unbroken, right? It's not degenerate. And then connected, CCC, TAA, CCC, TAA. So several instances. No one's going to say that this is a fusion. Here's the so-called fusion site right here. And so notice TTA, GGG up here, but you don't get another tandem repeat where you actually get an unbroken TTA, GGG until many sequences later. Here's another one. You can count them up. 1, 2, 3, 4. And then here's basically your fusion site where it connects to the CCC TAA. And so it's very broken. It's very degenerate. I've seen some critics look at sequences even before this and after this, and they've calculated a few more instances of these, but nonetheless, it's easily less than 20 of the TTA GGG and less than 50 of the CCC TAA. That's a very small signature if it were a fusion event. Yes, it's higher in density compared to the chromosome 9, but the chromosome 9 is actually more pristine. And so there's traits about the chromosome 9 site that are unique to itself. And then there's traits about the chromosome 2 site that's unique to itself, like the high density nature of it. But like you pointed out, it's fallacious to now say, well, it's high in density because it's the ancient remnants of a fusion event. Well, wait a minute. We don't have another version of Homo sapiens to look at and compare the genome and be like, hey, look, this is an example of a fusion site. And here's an example of a fusion site. No, in the same way, we don't have another universe to compare this universe to say, oh, look, this one looks old compared to the other universe, which has maybe traits or characteristics of being young. We have nothing to compare it to, but we do have examples of fusions in other organisms like pigs and equids. And from my understanding, they comprise a very large signature. I mean, what's a bigger signature, Brian? 38,000 base pairs or 798? Right?
30,000, yeah, bigger.
Exactly. Now we're talking. But 798, that's pretty flimsy. And then the authors of a very prominent chromosome 2 paper, anyways, what did they ask? They asked the question, why is this area so degenerate? And so even they recognized that. But anyways, go ahead, Brian, and maybe...
Well, Erika kind of just changed her argument once you pointed this out and said, well, maybe you could find some examples of it. But that's not what she said. She claimed that you could never find an example of it. That this means that because it's a head-to-head sequences and repeats, that automatically that means that a telomere's fusion must have happened. And then when you sit there and compare it to other genomes, it's like, no, wait a minute, you can see that in other places. And so then they
Right.
try to add the density and then say, well, maybe you could find that somewhere else. So it just kind
Right.
of like they're just kind of changing the argument as they go forward in order to not to get their catch with the
Shifting or
tail
moving the
between
goalpost.
yeah, catch with their tail between the legs.
If that was
Going
the challenge and the high density nature of it, so it's circular reasoning. Basically, they want to say, take the fusion site query, the 798 base pairs, and find that exact sequence anywhere else in the genome. You don't find it. It's like, well, no, duh. I mean, you're assuming that this 798 base pair region is a fusion, and now you want us to find that exact signature somewhere else in the genome. No, that's a unique signature. We find it in the chromosome 2 area, but hey, here's another similar kind of signature in chromosome 9, not exactly the same as the chromosome 2 one, but similar characteristics. It's in a head-to-head organization.
You
It's
probably the only way I could see that they could be able to prove this is that they could find an observed telomere fusion happening in a normal circumstance, in a normal situation, and in doing so, degenerate to 798 base pairs. But the degeneration of might be in, I can't remember, but the fusion site was oh yeah, what was it? Can't remember. Was it spiders, ants, something like that? But it was they said that when they degenerate, it was like 100 base pairs. It degenerated like whenever they saw telomere fusion, it degenerated a little bit, but it was like 100 base pairs less or something like that. It didn't really degenerate much. It was like a really, I mean, it's in order to, I think you even brought it up before, that in order to degenerate, you're talking about thousands and thousands of base pairs that would have to degenerate. That's a lot. And then you got the same problem with the centromere, the deactivated centromere having to degenerate. And that's even more because it's 250,000 to 5 million base pairs long. So, so that's a lot of degeneration. And so the laboratory, I think it was with the laboratory experiment that was mentioned, maybe with the mice. I think it was actually. Is it okay if I share my screen real quick? Because I think I know where it is.
Sure.
Okay. You should read my argument here. It's actually pretty good. But I think I put it in there with the one I said about mice. So I can find it. Fusion don't occur in the past.
Well, I saw there you pointed it out, right? The human telomere sequences are between 5 and 15,000 bases long.
Yeah.
So basically, if you were to fuse two together, you're looking at somewhere between 10 and 30,000. And so even if it did degenerate, how come we're not finding something like 2,000 base pairs in terms of a signature? Why is it less than 1,000?
Right. It's so much more less than what they are claiming.
I mean, you can give it, you can say, okay, you had a lot of degeneration. Let's say you went from 30,000 down to 2,500, and then more degeneration since the split. So now you're down to 1,500. I mean, even their best case scenario is still nowhere close to 798 base pairs.
Here's the one with the mice. To generate a mouse deficiency for telomerase activity, we delete the copy of the mouse telomerase RNA GNA genomic southern blots, probably with the transcribed region of the mouse telomerase RNA gene. This is from that particular paper. Identify single band, suggesting that EMTR in a single copy gene, data not shown, to delete the MTR gene from the mouse germline. We constructed the plasmid PMNT. I'm copying this directly from that paper. So, which allows replacement of the entire MTR gene with the gene produced using the standard protocols, WW6 mouse embryo stems, cells. Okay, I think this just tells what they did. So let me go. Okay, telomere's activity is thought to be required for telomere length maintenance in mammals, as in yeast. In humans, primarily fibroblasts that. Oh, this is it. Fibroblasts that lack detectable telomerase activity. Telomeres shortened by about 100 base pairs per cell division. 100 base pairs is not a lot. So when they degenerated it, it just degenerated by 100 base pairs per cell division. And that's with the, see, make sure that there's nothing about the experiment with wins. I went several times and he released Tompkins in Nature. So the lab experiment was okay. However, there's another problem associated with the lab results. So this is the lab results with the mice. Did you see that?
Yeah.
So yeah, that's 100 base pairs is how much it degenerated per cell division, it said. It's like, wow, okay. Don't they need like thousands to
Right.
degenerate?
I would need to ask clarifying questions, right? Okay, per cell division. So how many rounds of cell division are we looking at? So 100 per cell division, you know, I'd have clarifying questions for somebody like Ruhif on that one.
Right.
Because 100 base pairs doesn't seem like a whole.
No. That's what I was sitting thinking. I was like, it's not a whole lot there. 100 base pairs. But they would have to have, I mean,
But if you have a lot of rounds of cell division and it's 100 basically being degenerated per cell division, but there's a lot of rounds of cell division, then it could add up to a lot. Again, it's kind of vague. I would need to ask somebody like Ru. I'd need to ask clarifying questions.
If you had like 10 cell divisions, that's like 1,000.
It seems like they're really grasping.
Yeah.
Because they never predicted the degenerate nature of the fusion site. Now, if they said something, you know, once they made the prediction, before they got the data, if they would have said, and we expect whatever signature we find in the human genome to be very degenerate, a degenerate form of healthy telomeres.
All right.
Then you know what? They might have a case because they predicted it before they discovered the data. Now, because they discovered the data, they asked, why is the area so degenerate? And they have many different explanations. Okay, many different stories, I should say, but they're retrofitting the data. This is after the fact explanations.
All right, let's go ahead and move forward because I know that that we've been going on this a little bit longer than we intended. All right, let's share screen again.
What is it? Telomeric repeat. It is, someone just tell me. It's GGG, TAA, and CCC, ATT. Whatever. It's a telomeric repeat. I just did a video on this.
I think she butchered that one.
What was that, brother?
I think she butchered that one.
Yeah, her live audience is like chewing her chops on different things, but they support her overall views.
Right.
There's telomeric repeats that as a motif can be found in forward and in reverse in high densities in the middle of the chromosome if the fusion happens. That's the prediction. And secondarily speaking, there should be a second centromere, right? Because a chromosome has one centromere. So if chromosome 2 is the fusion of two ancestral chromosomes, there should be two centromeres. So there should be a telomere calling card in the middle of the chromosome and two centromere calling cards. These are alphoid repeats. That's what they're known. And so we have found this in like staggering detail, right? We have a 798 base pair fusion site located in the middle of human chromosome 2 that is degraded telomeric repeats. The fact that they're degraded is super important because telomeres can't experience, chromosomes can't experience fusion with pristine telomeres. They would prevent fusion. So the fact that there's degradation going on means telomeres in this ancestral condition were absent or failed in some way, resulted in a fusion site and then rapid degradation on top of that. So you've already got subtelomere regions, kind of like, worse versions of telomeres with regard to the sequences. So we found the fusion site, 700 base pair, 798 base pair long series of telomere.
Do you want to say something on the Centromere reference? Or did you already basically address that during the go?
So firstly, her best argument she's going to make soon, it has to do with the DDX11L2 gene and how apparently it belongs to a family of pseudogenes, the DDX11L family, I believe it is. And these genes are apparently always found next to telomeres. But this specific gene is found in the middle of chromosome 2, not next to a telomere in terms of the end caps. So the question is, why is it found in the middle of the chromosome? That's probably one of their better arguments that I'm ready to demolish. But with the centromere one, yeah, I'll share my screen real quick just to respond.
Well, let me
But
stop
we did-
share real quick.
But we
That's
did
fine.
engage it quite thoroughly
Yeah.
in our last show together.
Yes, we did.
But let me,
It doesn't hurt to repeat some of this because
Right.
might not have seen that video.
exactly. So for the audience, I kind of want to hit all of these important points. So firstly, we pointed out earlier that real functional centromeres basically exist in sizes of about 250,000 bases. What we find here, this signature is only about 41,000 bases long. It's got interruptions in it that are not centromeric like. And the so-called fossil centromere is situated inside a large functional protein coding gene. And so it's difficult to imagine basically how a so-called fossil centromere, this centromeric DNA, could exist inside a functional gene, a protein coding gene. But here's a few points that I do have on the so-called fossil centromere. Basically, as she said, you would need, if there's a chromosome 2, you'd have 2 centromeres. And so one would have to be deactivated. So notice here, another serious problem with the alleged cryptic centromere, okay, this is aside from the points I just said, is that its human alphoid repeat DNA sequence does not closely match homologous chimpanzee centromeres and chromosomes. So it seems like the homology isn't all that good. In fact, the differences in human versus chimpanzee alphoid repeat arrays are so profound that Half and Willard, in a later publication, so this is from the conventional literature, and they said this implies that the human chimpanzee sequence divergence has not arisen from a common ancestral, and then it goes into these repeated sequences, basically. Notice this. Of particular interest to the idea of a cryptic centromere is the fact that human chromosome 2 had 6 internal regions bearing alphoid repeats outside the centromere. Clearly, the alphoid region at the alleged cryptic centromere site is not a unique or an uncommon feature. So basically, you find other instances of these centromeric-like repeats, this alphoid DNA, in other parts of the genome, not just the chromosome 2. So this Jeffrey Tompkins here says 13 other human chromosomes also showed multiple regions of alphoid repeats outside the centromere. So here's an example. Basically, here's all the hits that you get of these specific alphoid sequences. So you can find, chromosome 4, looks like it's got quite a few. Chromosome 2, of course. Notice how chromosome 2 is your second largest chromosome. Right. So some people might say, oh, but you find the largest amount of these sequences in the chromosome 2. But at the same time, it is the second largest chromosome in the genome. And so
Well,
I don't think.
Would you say would you say that has more of an opportunity for these alphoid repeats to actually happen because of how large it is?
Yeah, it sounds like there would be more room, more space to have a lot of these repeats, and that goes for the internal telomeric repeats too. But you can see there looks like there seems to be quite a few on chromosome 4 here, quite a few on chromosome 10, chromosome 5, quite a few on chromosome 2. But again, I think the biggest argument against the so-called fossil centromere, Brian, is the fact that it's overlapped by a functional protein coding gene. So your 798 base pair region, that's overlapped by a highly functional non-coding RNA helicase gene, right? But this signature is overlapped by a protein coding gene. And so this tells me that these are not fossil remnants of an ancient fusion. They're functional DNA elements within human genes. And so there's a connection there, and that connection is probably for functional purposes.
Right. All right. Let's continue so we can get through this. Could you
Yeah, you know what? But before you continue, I wanted to really show the audience something. This was something that I have challenged the evolutionists on in debates. Okay. You know, I've done about 100 formal debates, but I've also engaged in a lot of just impromptu open mic debates, as you know,
Right.
Brian. So I've engaged evolutionists hundreds of times, and I'll typically show them this. This is the telomeric repeats. Notice we find these internal telomeric repeats, Brian, all throughout the genome. Okay.
Yeah.
Now, most of the time, they're just in isolation. Okay. But other times, like in the chromosome 2 area or in the chromosome 9, we find them in a head-to-head organizational pattern. But that's not the point. Brian, look at the chimpanzee genome. What do you notice? Well, I'll tell you what I noticed.
Like a
There
bomb.
is these internal telomeric sequences, they're generally absent in the chimpanzee genome. Notice you barely find them. But in the human genome, you find them all over the place. And so that tells me maybe these internal telomeric sequences, this is an active research project, these interstitial telomeric sequences, maybe they exist in high number in the human genome, but not in other primates, okay? Because there are unique aspects of being a human, right? Our higher cognition. We're a sophisticated species, basically. There's a lot, I mean, you can look at similar genetic sequences, Brian, between humans and chimpanzees. And although they're similar, there's actually differences in the way they're regulated or the way they're expressed. And that goes for sequences associated with the brain, because we have higher intelligence. We have that higher cognition. So I think these internal telomeric sequences exist in a high amount in the human genome for functional reasons that make humans unique, and chimpanzees don't have very many. And so I think that's an important point for your audience to take away.
Would you say that that God could have created humans with far more variations than what we see like in primates, where primates have variations, but they may not have it as much as us. And so we would expect a far more variations between human beings than say between primates. I think it's I think I'm working off the idea of what your friend Matt mentions on the you know, the differences within humans, like we have, you know, we don't seem to have a whole lot of we don't variate to the point where we look completely different, but yet we have enough where we can, you know, where the human race can scatter across the planet Earth and wind up like different skin colors, different facial features sometimes. You there?
Yeah, that's a good question. I mean, if I'm understanding your question accurately, humans do have very low variation. So humans are all about 99.99% the same, which makes sense because the book of Genesis says what? That God created two people just thousands of years ago. And so if that's the case, humans start off as two, Adam and Eve. What does that do? That restricts levels of genetic diversity. You're not going to have a whole lot of genes floating around. But animals, God didn't create just two. God created animals in large populations in fully working ecosystems. And so we'd expect animals to have a lot more variation than humans. And that's actually exactly what we find. We find animals mostly have a lot more variation in terms of DNA variety than humans do. And I would argue that's confirmation of the account of origins in Genesis. I mean, chimpanzees have a ton of genetic diversity within themselves. Humans, not very much. But to your point, I've got paper after paper here. I've given whole presentations on this, where humans have massive differences in the way their genes are expressed. Notice this, 90% of the human genes were turned on at significantly higher levels than chimpanzees, or these similar genes are regulated differently. There's differences in alternative splicing, differences in epigenetics, you know, and here's all the papers that people can look to. The 3D genome. is distinctly unique in humans. And again, those internal telomeric sequences, we find them in huge amounts within the human genome, and we barely find any in the chimpanzee genome. And so there are parts of our genome that help to make us unique. And I think those interstitial telomeric repeats, I think those are a type of DNA element that does make us unique, Brian.
Awesome. All right, let's move forward. My legs are getting stiff.
We found the second cryptic centromere, and it maps to chimpanzee 2B. When I say it maps to it, I mean it shares the genes upstream and downstream of this proposed cryptic centromere that map to the genes around the normal chimpanzee 2B centromere. Those two things map, right? So we've got both of those, and then moreover, the most important thing, and in my opinion, is the overall synteny. The chimpanzee 2A and 2B share 98% approximately percent similarity with human chromosome 2. And I know this because I tested it, and you're going to get this in a human chromosome 2 video that's coming up. But what I did is I did a comparison of chimpanzee 2A and 2B to human chromosome 2, and I did full coverage. Every single base pair is covered. So
is that correct?
So we could go down a very long rabbit trail on Synony, because there are some strong points. Christopher Roop has pointed this out as well, that there isn't very good synteny. There's portions of your chimpanzee genome in and around the area of the chromosome 2 where they infer a lot of similarity, but it's actually not sequenced enough, or the data is not reliable enough to make a conclusion. But it's one of those things where it's agnostic. So for sake of argument, let's just say there is synteny, Brian. And for anybody unfamiliar with it, is basically shared gene order along chromosomes. Okay. But I would argue that synteny and the 3D organization of genomes in general, let's say of, humans and chimpanzees here, is best explained through common design, because they've discovered, and there's paper after paper on this, I've cited many in my presentations and articles, the way that genomes are organized. Okay, so the ordering of these genes within chromosomes, these similarities, they're there for functional reasons, because chromosomal order actually influences the way genes perform, the way that they function. And I like to point out the elegant and sophisticated nature of the genome, the way genes are organized, is stronger evidence for design and not common descent. So sure, if there's synteny, well, synteny exists for functional reasons. And so that would be an agnostic line of evidence. That could be explained through descent, as they would, or common design. So for sake of argument, it doesn't help them, Brian, is my point.
Right. It's also like the confirmation bias, where you just kind of don't consider what the other people say as well. But there's also a kind of a circularity in that type of argument. Like the base pairs are matching here, so the base pairs are matching there, so they must be a telomere fusion. Well, yeah, we're gonna have similarities, and you only have like these four chemicals, these four bases that are being reordered in our DNA to spell out information or message. And I always find it really fascinating, the fact that there's only four chemicals, but yet these can be spelled out in such a way and ordered in such a way that it actually spells out a message within our genetics and within our DNA, and so much functionality as a result. Yeah, that always amazes me at that kind of complexity. But in any given case, you're obviously going to find some sections within the genome that's going to be the exact same base pair alignment as you would find in chimps or even in other animals. You could probably do the same thing with mice, between mice and humans, and find base pairs. It doesn't mean there's a chromosome fusion between us and mice. It just means you found the exact same base pairs in the exact same spot, and that's going to happen on occasion, because you just got these four chemicals. And often when we see the sequences, We see them, like you said, they spell them out like in letters, like AT, GC, that sort of thing. They and like, what was it, Adenine,
Adenine, yeah.
Thymine, or something like that. Trying to think of the bases.
ATC, so basically adenine, thymine, guanine, cytosine.
Yeah, that's it. And so they just kind of spell them out into the form of letters as part of that sequences of the DNA, because DNA is, it's double stranded too on top of that. But in any given case, so you kind of can expect base pairs to be the exact same. If you were really looking for it, you're going to find base pairs that's
and gene order too, right? So
Yeah.
Synony, it's not a surprise. I mean, yeah, you're going to find similarities in gene order, and we would predict as creationists that these are there and organized that way for functional reasons. And so we go in looking for function. These are testable predictions we can make. And the trajectory of discovery favors near genome-wide functionality.
Right.
And so whether it's hierarchical patterns, whether it's syntony, homology in genetic sequences, they're there for functional reasons. I like to say these are hierarchies by design and not necessarily descent. See, the differences make all the differences, right, Brian?
Right.
We could look to similarities all day. We can look to syntony all day. But what about the differences? What about the Y chromosome dissimilarity? It's only 35% the same between humans and chimpanzees when you consider overall architecture, gene content, size differences. And so why don't we talk about the differences that they can't account for? Differences in gene expression, alternative splicing, gene regulation, epigenetics, the 3D organization of the genome. These are discriminatory lines of evidence, Brian, that they can't explain. The similarities are easy to explain. Common design. They'll explain the similarities due to homology. But the differences, they really struggle with, brother.
I think, I can't remember if it was Dr. Jensen. I think it might have been Dr. Jensen. But they presented 3 different types of experiments that scientists have. And one type of experiment is when basically both sides, both conclusions would have accounted for the same data, in which case you learn nothing, because essentially we're from both perspectives we can get the same results, but it doesn't really do any good, because you can get the same results if you consider the other side as well.
Well, there's a lot of overlap in the creation versus evolution world, and so there's a lot of observations that both models can explain. Homology, it would be like me saying, Brian, creation's true because the sky is blue. Well, wait a minute, evolution can be true if the sky is blue too. Both models can explain the data. And so because there's a lot of overlap, we have to use experiments, whether it's a type 1 experiment, type 2 experiment. I like to say we need to look to discriminatory lines of evidence that only one side can explain.
Right.
And a lot of these differences are just that. Discriminatory lines of evidence that only creation or the separate ancestry model can explain.
Right. Are you finished, or do you want to continue?
You know what, to leave no stone unturned on this syntony argument, also the homology argument, share my screen real quick. Okay. So right here, you'll notice Contested Bones, highly recommend, excellent book. And so they point out, at least when it comes to homology, the sequences that flank the reputed fusion site should match up with the sequences near the ends of the corresponding chimp chromosomes. Unfortunately, we cannot easily test this. This is what I was saying earlier, because the ends of the 2 corresponding chimp chromosomes have not yet been fully sequenced. So there's a lot of inference going on in terms of what those sequences are. However, this is a fascinating point right here. The actual region that contains the reputed fusion site, which is assumed to derive from 2 ape-like chromosomes, now appears to be DNA that is uniquely what? Human, and is absent in all ape genomes. The UCSC genome browser shows that all the great ape genomes fail to align with either the so-called fusion site or the flanking 3,000 plus base pairs. And they point out this is a very serious problem for the fusion hypothesis. This excerpt here says this entire region of human chromosome 2 does not appear to have homology with any part of the 2 chimp chromosomes that are thought to have fused. And so specifically, they're talking about the much larger 200,000 base pair region that flanks the so-called fusion site. And so it appears that the homology specifically is limited. And it also sounds like the Synony is inferred mostly because of incomplete data. But even if that wasn't the case, again, Synony exists for functional reasons. And there's many papers demonstrating that. So Synony is a very weak argument.
Hey, Donnie, let me ask you about the solid DNA. That was something on my mind a little bit ago. I was going to ask you about, but since your quote automatically referenced that, my understanding is that there's a lack of solid DNA in these fusion sites or something of that nature. Is that correct?
Good point. I like to, when I engaged in an impromptu debate or discussion with Dr. Dan Stern-Cardinal, this was something I focused on because remember, overall, the chromosome 2 argument is, and you pointed this out too, brother, and I appreciate it, it's agnostic to the debate because if there was a fusion event, okay, according to the evolutionary model, this would have been a near extinction event. Basically, the person who experienced this fusion would be the ancestor, the single ancestor of every single person who exists on the planet today. Every single human being has 46 chromosomes. So where's the humans that have 48? No, they must have all died off. Okay, so if the evolutionary model is true, they'd have a very difficult time explaining how this unilateral mistake, this fusion, could become fixed, stuck in place, where now everybody has 46 chromosomes and nobody has 48. Basically, all lineages or all tribes on the globe at that time would have eventually had to have been wiped out or killed off in order for that fusion to proliferate. So if any model is going to explain the data, if there really was a fusion, it'd be the biblical creation model. We have 2 bottlenecks. We have a bottleneck of 8 people at the flood. And then we also have a bottleneck, somewhere between a couple hundred years after the flood with the Tower of Babel, somewhere between 10 and 150,000 people. And so before people spread out to all parts of the globe, that chromosome fusion could have proliferated throughout the population. I'm not saying that's the case.
Hey,
I don't believe that's the case. I'm just saying worst case scenario, if there was a fusion, the creation model explains it better anyways. So the evolutionists are kind of stuck between a rock and a hard place, right?
I try to explain that to Ruhip as well, because he didn't seem to understand the argument. But in the process, when I tried to explain it to Ruhip, he said, well, did pigs experience a extinction event and that sort of thing? How would you respond to someone who brings up the pigs in reference to that particular argument that they wouldn't have experienced an extinction event? So if a fusion, but I think that they misunderstand that particular argument in the process.
Yeah, he doesn't understand basic population genetics and just how fixation works. Yeah, that specific pig species has a fusion, apparently, the chromosome 6. But there's other very closely related pigs that have differing number of chromosomes. So no, that fusion did not proliferate or become fixed in all pigs.
Wow, you said the exact same argument I made, and I didn't even hear that from you. So I apparently understood the argument. I mentioned that different pigs have different chromosomes, and so that would not work at all. And I mentioned that in my argument as well. So you said the exact same thing. And I've never even collided. I never even talked to you about this as far as what we have said till now. So yeah, you said the exact same thing I did.
Right. It's almost common sense, right? Great job. Right. Yeah, there's different pigs that we all agree are related, but yet differing number of chromosomes. So no, it wasn't fixed. But in humans, guess what? Every single human people group, no matter how divergent they are, even if it's your most isolated sub-tribe, that's just completely isolated from the rest of the human population, guess what we all have in common? 46 chromosomes. So that fusion would have had to become fixed. It's not like, 75% of humans have 46 and the other 25% have 48. No, it's literally fixed. That mistake's been stuck in place. And so that's very difficult for the evolutionary model to explain. A fusion has not been completely fixed in all equids. There's not been a fusion that's been completely fixed in all pigs. No, there's differing number of chromosomes. So you nailed it. Yeah, you gave the right answer, brother.
Yeah.
So
Oh, go ahead.
well, I was going to say for the satellite DNA.
Yeah.
So this paper right here, see if I can pull it up in time. Basically, there's a type of satellite DNA, ape-specific satellite DNA, that doesn't exist around human chromosomes, but it does exist in like chimpanzee genomes. It exists in gorilla genomes. I'm trying to find, oh, here it is, finally. If you ask the questions in the order that I have my slides in, it makes it a lot easier. No, I'm just kidding. That's a line Kent's been using for years. So, okay, so here it is. This is the famous Ventura paper, I like to say. So notice how they point out chimpanzee and gorilla chromosomes differ from human chromosomes by the presence of large blocks of subterminal heterochromatin. So this is all very technical. Heterochromatin, basically these very dense pieces of DNA comprising a lot of repetitive sequences known as tandem satellite sequences. And so this specific kind of DNA is not found in human chromosomes. There's another difference, but it is found in chimpanzee and gorilla chromosomes. Okay, if we were related to chimpanzees and the other primates, then why are we completely missing this type of satellite DNA? And that goes for the chromosome 2. It's not found around the chromosome 2. It's not found anywhere. So the evolutionists have to argue, well, this kind of DNA must have been lost over time. Or maybe it's been added in the chimpanzee and gorilla line, you know, but the point is it's not there in humans. So if there was a fusion, because we lack this satellite DNA, this kind of satellite DNA, Brian, the best explanation is that it is a human specific fusion and it's unrelated at all to chimpanzees, to gorillas. And so that's another argument for the biblical creation side. I'm glad you brought it up.
Awesome. All right.
Human and ape chromosomes. They also found some problems with it as well. But anyway, so that's what led to the whole idea of it. And then they thought they found in the early 1990s actually a DNA sequence that represented the fusion site, which they claimed was a head-to-head fusion of human telomeres. And so it was a very small signature. It was only about 800 bases long. And the reason I say small is because human chromosomes are 5,000 to 15, or I'm sorry, their telomeres at the ends of them are 5,000 to 15,000 bases long. So if you have two telomeres allegedly fused together, you should have a signature of 10,000 to 30,000 bases.
No, you shouldn't, because remember, degradation of the telomeres is like a prerequisite for fusion to occur. You wouldn't get pristine head-to-head telomeres in like the same identical densities to those at telomeres, because if those densities existed, the fusion wouldn't happen. Jeff is a geneticist.
Okay, what is she arguing here?
So I pointed this out in our last talk, but she's essentially arguing that since healthy telomeres help to prevent end-to-end fusions, both of your two fusing chromosome tips, they must have been degraded prior to the fusion. So essentially she's saying the telomeres would have eroded to near extinction. Okay, but I would argue this seems unlikely to be the case, that these telomeres were degraded to exactly the same point, because that would coincidentally yield a minimized and semi-symmetrical fusion site. And so that
I would probably question whether, I mean, what exactly is she comparing it to determine that it would be near non-existence to be degraded to that point?
Right.
It's not based upon anything I know of, unless she's just kind of arguing in a circle.
Well, and then basically that would almost completely remove any kind of telomeric signature. You know, it'd be like, okay, well, that evidence isn't there because they basically almost nearly broke off. But again, this seems to be a rescue device after the fact, because like you said, what are we comparing it to? Are we comparing it to a fusion that happened in another universe in a human population that, you know, it didn't experience this kind of erosion before the fusion, and so now we're comparing the two? No, we see other species like in the pig, chromosome 6, where they have a large signature. So why didn't their tips erode to the point where the fusion was capable? No, we find in nature fusions happening, and we find fairly large signatures. So, you know, again, why did the authors ask the question? Why is the area so degenerate? They should have said the area is degenerate, and this is what we expected, because the only way for a fusion to take place is that the tips needed to be eroded to near extinction. No, they didn't say that because they didn't predict it. If it was that easy and that simple, they should have predicted it, Brian.
Right. They're claiming that after the fact. And
Right.
that's a problem, because it's like they're kind of like saying, okay, we can't claim a fusion event unless we degrade it to near extinction. and
Well,
then so they assume that happened that way.
It seems very far-fetched. Okay, they want us to believe that both of the fusing chromosomes were degraded to exactly the same point, creating a minimized and semi-symmetrical fusion site. That's just too much to swallow.
Right. And thousands upon thousands of base pairs being degenerated.
Right.
And no evidence at all that happened in the past. So
Basically the same point on each side where they can come together nicely for a fusion.
Right.
Almost like it was preordained by the evolutionist god time.
Right. All right, let's continue.
800. And so on top of that, the fusion site is very muddled, if you will. It's evolutionists would call it degenerate. In other words, it's not a pristine head-to-head fusion. It's only about 70%, similar to a pristine fusion of the same size, 800 bases. So anyway, so it isn't that clear cut. And on top of that, the fusion site is actually found inside a long non-coding RNA gene. And if things couldn't get any worse for the evolutionary fusion story, the fusion site is actually a second promoter or a switch inside this gene. It binds RNA polymerase II, which initiates transcription in the middle of the fusion, so-called fusion site. All sorts of transcription factors bind to the fusion site. And I basically mined all these ENCODE data sets to verify all of this.
So
And if you look on a
again-
genome browser, you will see the fusion site inside the beginning of this gene. So
So again, when he's talking about the critical nature, how often it's transcribed, how important it is in all these tissues, like again, it spans the fusion site in roughly 21% of transcripts, and it's expressed overall about as much as or about 5% as often as your average run-of-the-mill gene. So no, none of this has any bearing on the fusion site being a fusion site. But moreover, genes can also move around in the genome. So even if it was an absolutely critical gene that had an imperative function, it is possible that that gene existed in the ancestral condition as well and just moved over the now somewhat free real estate fusion site. That is also possible. But you don't even need that.
All right, Donnie. Two arguments here. One, she's saying that that she thinks what he's saying about fusion, about the being so small and pristine and things like that. She thinks that's irrelevant to whether it's a fusion site or not. And the second argument, which I know you're dying to respond to, is her argument about the gene moving over and to a functional gene moving over to that, because I know you responded to that before.
Yeah, I find that specific one to be a whole lot of imagination, inference. Okay, well, maybe the gene moved over the site, and now the site is essentially acting as a promoter for the gene. I'd like to see some empirical and experimental evidence for that. Okay, because firstly, the fusion site region comprises multiple start points for transcription where RNA polymerase II binds, as Dr. Tompkins has pointed out. It's an amazing source of genome activity, but apparently this all has taken place due to genes moving around and fusion events taking place and just all types of messiness going on in the genome. No, the most parsimonious explanation is that the site is basically a functional component inside an essential gene. This gene is on fire. It's highly expressed, and I should probably share screen because because they like to attack, as you know, Tompkins.
Yeah.
Oh, this is just a Tompkins argument. Well, let me share screen. You can just look up the gene. Okay, notice here. Just look up the gene, DDX11L2. Click this website and there's a ton of different drop boxes showing what it's expressed in, what it's co-expressed in. And so DDX11L2 has over almost 2,000 functional associations. And then they'll try and say, well, it's expressed at low levels. Well, and then they want to compare it to protein coding genes and their expression levels. Okay. Well, that's apples and oranges. That's not apples and apples. We understand now that when it comes to a lot of your non-coding genes, you have to look at co-expression. You have to look at a lot of different factors, because non-coding genes that are important, RNA genes, they could be highly functional, but expressed or transcribed at low levels versus a protein coding gene, because they function differently. And that's why your protein coding genes tend to be more similar across species, Brian, because they tend to be the building blocks of creatures. Okay. But your non-protein coding genes tend to be more species specific, as they provide the metadata for that which is the protein coding genes. Now, Dr. Tompkins responds here to this argument about how some transcripts don't have it spanning the fusion site. And so notice here, one of the main proponents of the alleged fusion site as evidence of human evolution is Ken Miller. He's the one who first made this argument. Miller initially claimed that I was wrong about the fusion site being situated inside the first intron of the DDX11L2 gene, based on data that excluded the 3 exon version of the gene. I then conversed with Miller via email, and he did concede, notice this, he did concede that when one includes the longer transcript variants, the fusion site is in fact located inside the gene. Now, Miller, like Gutsick Gibbon, they got their rescue devices. So he attempted to explain the longer transcripts, because now they have to explain. So yes, it's in the first intron and it does span the fusion site. So now they have to explain the longer transcripts where that is the case. So he claimed that they are easily explained by variability in the termination of transcription, so that occasionally a somewhat longer RNA is produced. Tompkins debunks this. This argument is completely erroneous, because the direction of transcription for the DDX11L2 gene, which is encoded on the minus strand, is the opposite of what Miller is claiming. The longer transcripts begin ahead of the fusion site at the 5' end of the gene. It is a well-documented fact that the transcription begins at the 5' end of genes. And so, he goes on to point out why this is not spurious. There's a lot of function associated with it, and their only argument really is that it is spurious. Notice this. There's one argument I wanted to point out about the right here. The fusion site sequence binds to at least 12 different transcription factors, including RNA polymerase II. We pointed that out. That's a key enzyme that transcribes genes. It's actually co-expressed with many other important genes. And so there's no way anymore that you can say that this is spurious. And you can just go look into the database yourself, as we've pointed to. It has 2,000 functional associations. So I think that essentially debunks her argument. But yeah, the gene moving over, that's a lot of guesswork. Maybe this, maybe that.
Let me ask you a question with that.
Yeah,
I was going to ask a question. Do we ever see genes moving like that usually?
Well, there is translocation. So, genes can move around for sure. We know that ERVs and other classes of retrotransposons. Actually, ERVs seem to not move around anymore, but there are jumping genes that move around, elements that move around, turning genes on and off. And so, you know, there is the relocation of DNA elements and genes in the genome, but to take that and extrapolate it to where this DDX11L2 gene just perfectly moved across this site, and now there's this elegant connection between the site, right, the active promoter, and the gene itself, where basically without that site the gene couldn't function. Without that gene, the area couldn't function. No, there's sophistication going on here that it's not very parsimonious to say that this gene moved across the site. Does that make sense, Brian?
Yeah. Is the surrounding genes surrounding this also functional?
Oh yeah, it's a hotbed of genetic activity. There's many functional genes.
So you'd have to
the
have
area.
that gene moving pretty good distance to, I mean, of course, technically speaking, this is kind of circular also, because this assumes, like in the case of a pseudogene, that all pseudogenes are non-functional and that sort of thing, because they don't know what the function is of everything in our genetics.
I think what they'll try and say is you basically have, you know, let's say you have 3 genes. And I'm kind of just guessing here, but you have 3 genes, and the DDX11L2 gene is at the end. Okay, so it's not over the fusion site, but then you get a gene deletion. Let's say the middle gene deletes, and then your gene at the end, the DDX11L2, moves closer because the middle one's removed. So that one connects with the first one. And now that happens to be over the site. You know, so by move, that might be what they mean. But nonetheless, it's still very unlikely that that gene moved over perfectly across this site,
right,
that is now an active promoter for the gene that happened to move over the site. So, you know, that's probably how they'd explain it.
Does
But
other evolutionists also believes that the gene moved, or is it that just an argument that's isolated to Erika?
yeah, they're just trying to say, they're trying to make a case that even if it's functional, because in all reality, there's no way to argue that it's spurious anymore.
Right.
I mean, the data is too strong, suggesting that it has activity associated with it. So they're just trying to cover all bases by saying, well, even if it is functional, it could have just moved there from another location. So, you know, they want their cake and eat it too, basically.
Yeah. But then again, a lot of them will claim it's a pseudogene. So this kind of seems to be self-contradictory to how they view the DDXL11L2 gene.
Yeah, that's all based on evolutionary presuppositions. They assume that, you know, there's these non-coding RNA genes that look similar to protein coding genes. And so, you know, they must be broken versions of those protein coding genes, right? This is a genetic mistake. But no, we're finding out that so-called pseudogenes have important functions. You know, your pseudogenes, they're discovering, actually help regulate their protein coding counterparts. And so they're necessary for the protein coding versions to function. And therefore they require the similarities to the protein coding counterparts. It doesn't mean that they were once a protein coding gene that was hit with a mutation. Maybe it deleted the promoter, you know, something went on that destroyed it, and now it's non-protein coding. No, it's actually designed that way. It's necessary to sustain healthy life processes in the cell.
Right.
And so when they say pseudogene, that's an evolutionary assumption.
Right.
Because it's not a functional gene. It's actually categorized as a pseudogene. Now let's see if he actually tells us the real punchline of this gene. Which, if you know the real punchline, say it in the chat now.
Any genes in the human genome have multiple promoters or these switches. A lot of these switches are in front of the gene, and a lot of the switches are inside the gene right at the beginning, maybe after the first exon, like the fusion site is. So anyways, it is actually a functional feature inside a gene. And this gene is an important gene. I actually looked at the expression of this gene. It's expressed in many different tissues in the human body. It's connected with the expression of other genes in top-level networks, including the formation of blood cells. And I actually annotated the messenger RNAs that were produced from this particular gene that the fusion site is in. And these messenger RNAs are full of all sorts of RNA binding sites and all sorts of functional features. So it's not a devolution might say, well, it's just spurious transcription, you know. No, it's a real gene that's producing real transcripts that do real stuff, important stuff. And
it's a real pseudogene that is transcribed spanning the fusion site 21% of the time and is expressed about as often as 5%, or is expressed 5% as often as other genes. He's still not saying the punchline though. He's not telling you the most important
see,
part
Brian?
against 11.
Yeah.
So she's making that argument where basically it sounds like it, at least she's comparing the activity of a non-coding gene like the DDX11L2 gene to that of protein coding genes. But you can't properly compare long non-coding RNA genes like the DDX11L2 gene to protein coding genes, because long non-coding RNA genes are expressed at much lower levels. This is well documented in the conventional literature. And so the functional or biological significance of a gene, it can't be compared by comparing it to, well, look at this protein coding gene and look at how much activity is there, how much is expressed. No, you need to have a combination of data that includes, and Dr. Tompkins points out, it includes epigenetic marks, transcription factor binding, it includes expression, co-expression with other genes. And when you actually consider all of that, you can see that the DDX11L2 gene is highly functional. And those transcripts where the gene overlaps the site, which she keeps downplaying, okay, they haven't been able to explain how that's the case. Ken Miller attempted, but Dr. Tompkins refuted it. And so this isn't an apples to orange comparison. Anyways, I just wanted to point that out, brother.
Yeah. You mean it's not an apples to apples comparison? But
It's not
like
an apple, right? But
more like an
basically,
apples and oranges is what she's comparing.
Exactly.
Would she say 5%? Is that accurate, or is that only accurate with between coding and non-coding protein or protein coding or non-coding?
Yeah, it's kind of like my van. I have a front windshield wiper that I use very frequently, even if it's not raining, you know, shoot the water fluid and clean it off. My back windshield wiper, it works, it's there, it's functional, it's got a purpose. I barely use it though. Does that mean, so that can be, so your back windshield wiper can be compared to your non-protein coding gene like the DDX11L2. It's not used that often, but when it needs to be used, it's functional. Your front windshield wiper, you can compare that to your protein coding genes. It's used a lot more, it's expressed a lot more, okay? But both are still functional. It's not like my back windshield wiper's junk. It's functional, it has a purpose. And so you can't compare a long non-coding RNA gene like your DDX11L2 gene. You can't compare that to the expression levels of a protein coding gene. Like you said, brother, it's apples and oranges. Didn't
you also say that earlier that that the non-coding protein genes are also are required for the protein coding genes, that they're actually co-expressed with them? Is that correct?
Right. Your non-protein coding regions are basically regulating the function of the protein coding genes. There's a connected, see, your evolutionists, Brian, they have a very unsophisticated view of the genome. The genome is not just this linear set of letters, A, T, C, G. No, it is multi-dimensional. It's a 4-dimensional code that's chock full of meta information. The 4th dimension being time, basically. You know, and so there's a lot going on. There's a lot of interactions going on between your non-coding regions and your protein coding regions. And that's why with your non-coding regions, you have to look to co-expression, like you said. You have to look to epigenetic markers. There's a lot you have to consider to conclude, is this a functional gene or a non-functional gene?
Right. All right, let's continue, unless you got more to say on that.
No, that's good, brother.
Let's see if he says it.
And so the fusion site is bogus. It is a feature, a functional feature inside a gene. And so I can get really deep in the technical muck with this. I actually have an entire talk that I gave at the International Conference on Creation in 2018.
Brian,
where I actually developed some.
I have an idea. I have an idea if you think it's a good idea to really make sure we have enough time to hit her last two arguments. Did you want to speed up her video like she sped up yours? Maybe put it to like 1.5.
That's really
Because I have watched this over the last couple nights, so I do know what she's going to say. So I don't necessarily have to listen to it at normal. Maybe
playback
1.5.
speed.
Yeah.
So 1.5.
Yeah, 1.5 should be good.
All right, let's
You
see.
got it.
Software to scan the human genome for internal telomere sequences and then line up those genome coordinates with all these all sorts of internal telomere-like sequences that are associated with a variety of functional aspects and all sorts of things. So, anyways, so that they actually published a paper on that. The whole fusion scenario is not viable.
Tell them about DDX11L2, Jeff. Tell them about the gene family it belongs to.
When you begin looking at the genetic data, in fact, I even looked at the so-called tryptic centromere site, which is less convincing than the fusion site. So if you have two chromosomes fused, because each chromosome has its own centromere, theoretically you would get two centromeres.
He's gonna skip it. Okay, I'm gonna have to tell you guys the punchline since I guess Jeff isn't going to do it. And I watched this. I don't remember if he actually says it, so I'm going to spoil it for you. But to do so, we're going to need to hop back over. We're going to need to hop back over to our images over here. Okay, you guys. So DDX11L2, here's where it is, right? It spans the fusion site. Let's just take the best case scenario for Jeff, right? It spans the fusion site. It's here in the middle, located in the middle of this fusion site, this 798 base pair long sequence of DNA in the middle of human chromosome 2. It's a pseudogene that belongs to a family of genes in the DDX11L family. You want to know where every other member of this gene family is found exclusively? In telomeres. It's in telomeres, you guys. They're all found in the telomeres of chromosomes. Here. So what's a telomere-specific pseudogene doing in the middle of human chromosome 2? I ask you, you people, what is it doing there? Can you tell me? It's because it's a fusion site. It's a fusion site. And Tompkins got asked this question in a different video. Let me see if I can actually find it. It actually might just be worth looking at here, whilst we're already with one another. So let's see. It was in Logos Associates, I think. Tompkins. This is a different Tompkins video, but our friend Dan of Creation has asked a very interesting question about the telomere-specific pseudogene found in the middle. Let's see what he says.
It's from Creation Myths again. Why are all the other instances of the DDX11 pseudogene family in subtelomeric regions? And would you mind maybe explaining that for our lay audience?
Yes, I am aware of that they are in subtelomeric regions. Subtelomeres actually have lots of genes, and in them, and they're actually very, very important regions of the genome. Lots of gene activity going on.
Yeah, and what about them being found in telomeres? Why is there a telomere-specific gene in the middle of human chromosome 2, Jeff?
On there. No, I don't know why there's so many in those regions. Then you also have the protein coding DDX genes as well, which are not.
He doesn't know. He does not know, you guys. So I don't know what to do with this man. I don't know what to do with them. I'm getting so worked up about this. It's just, what do you do with this kind of information, right? Like Jeff is an actual geneticist, one that is highly lauded and praised by all these younger creationists who think non-geneticists don't get to weigh in when Jeff he can't actually have a conversation on the topic that he is debunking. And you can go back and he doesn't answer the question. You can go back and watch the rest of that, but we're here to watch a different video. So he's
All right. So a lot there. So essentially she's saying this is that the particular family of genes that this is part of is telomeric specific genes, that they're only found in telomere sequences. And when she asked Tompkins about this, or when Creation Myths, which we know who that is, when Creation Myths asked Tompkins about this in the Q&A, his response was that he wasn't sure, he didn't know why that was. What is your response to that? Is that the first argument you were talking about that you was going to get to?
Yeah. Firstly, I believe the DDX11L family, these genes, they are found not necessarily in telomeres, but in subtelomeres. And so close enough, but subtelomeres are basically regions of the genome that are immediately adjacent to your telomeric repeats, like your tips of your chromosomes, essentially. And so she's arguing that, and I've got some slides here, so I'll share a screen. She's arguing that since these genes are typically found at the end of chromosomes in these subtelomeric regions, why do we find the DDX11L2 pseudogene in the middle of chromosome 2 next to what they say are degraded telomeres? that fuse together, basically. Okay. So, I mean, admittedly, it's a decent argument. And I think they think it's kind of a fatal blow. I just engaged this point in a formal podcast debate I did with Base Theory, Grayson. And then we had an open mic that we do every 10 days. 2 open mics ago, I debated Grayson on this issue. Okay, so here we go. My slides addressing Gutsick Gibbon. So pseudogene family. Okay, firstly, this argument raises more questions than anything. I would ask, how do you know this gene, the DDX11L2 gene, yeah, they called it that. This gene didn't call itself DDX11L2. This gene did not put itself in the gene family, the DDX11L family. It certainly didn't call itself a pseudogene. That's evolutionary presuppositions, okay? If it's functional, if it was never once a protein coding gene that was broken and can no longer produce a functional protein, that doesn't mean it ever was that, right? It could be designed the way it is, so not a pseudogene. So my question is, should this gene be called DDX11L2? Maybe it's not part of the DDX11L family. That's a question. And if it is part of the DDX11L family, why? You know, they'll say, well, similar sequences, you know, nested within this family. But nonetheless, that would be my question. How do you know? And I have a feeling it would be based on evolutionary presuppositions, okay? Maybe, though, the gene is necessary to be near internal telomeric repeats. And since there's a lot of similarity to interstitial telomeric repeats, Brian, to your end caps, your telomeric DNA, then that makes sense, because your chromosome 2 has what? It has many instances of TTA, GGG, CCC, TAA. And so we know that these genes specifically do help with or function in chromosomal stability, architecture. And so for the function of those internal telomeric sequences, maybe they depend on that gene being near them. And so this DDX11L2 gene being near this site, maybe that's required for functional purposes, okay? A lot of this, again, is just raising questions, clarifying questions, because a lot of times the evolutionists, they won't know. When you cross-examine them and say, why is this gene part of that family? They might not even know. And their argument might actually be kind of weak, right? Like, well, there's similar sequences to other genes within this family, okay? I'm not saying this is necessarily the case that it doesn't belong in this family, but it's certainly a question that should be asked. So notice here, this is what Chris Roop and John Sanford say. While the expected subtelomeric repeats are not present in the region of the reputed fusion site, there is a gene called DDX11L2 immediately adjacent to the fusion site. This gene is homologous, this was her argument, right, Brian, to a gene family, that gene family is the DDX11L family, that is usually found in close proximity to human telomeres. At first, this might seem to support the fusion hypothesis, since this so-called site is found close to a gene typically seen near telomeres. However, we need to look more closely. A similar family of genes is also found in the chimp genome. While these genes are also generally found toward the ends of chimp chromosomes, they are separated from the telomere by a sizable amount of intervening DNA. Remember we discussed this earlier, Brian?
Yes.
The very same ape-specific satellite DNA described above. They talk about it earlier, this basically ape-specific satellite DNA that's not found around human chromosomes. Well, in chimp chromosomes, it is found. And so these genes basically being within subtelomeric regions or next to really close to telomeres within the human genome, that type of satellite DNA is not there. But in chimps, it is. And so there is a difference there for sure. I'd argue it's for functional reasons. Okay, so they say, so if we assume that the DDX11L2 gene on chromosome 2 came from an ape fusion, it should be separated from the telomere-like fusion site by this large ape-specific tandem repeat, but we don't find it. Absence of this intervening DNA between the DDX11L2 gene and the reputed fusion site is not consistent with the idea that a fusion occurred in an ape-like population. So basically, best case scenario for the evolutionists, if this really does imply that at one time the DDX11L2 gene was at the end of a chromosome next to a telomere, but now it's in the middle because a fusion took place. Well, again, the biblical creation model can best explain it because it is a hominin or a human specific fusion. And for it to become fixed in all people, it would be very difficult for the evolutionary model to explain how it could be fixed, but the biblical creationist could explain it nicely. But I have a brother in the Lord. He's helping me with an active research program, Craig Brown. He's doing great work, and he's doing a genome-wide search of these genes within this so-called family to see if it really is the case that these are only found next to telomeres. And his research seems promising. He's already told me that he seems to have at least located one that's more so next to centromere DNA and not necessarily next to a telomere. And also that it appears it might simply be the internal telomeric repeats that these genes need to be next to, because there's a coordinated function going on. There could be a function in chromosomal stability. But anyways, it's an active research program. I don't think the argument is as strong as they're making it out to be. Clearly, it's a decent enough argument because Jeff Tompkins, he didn't really have an answer for it. But again, I think we have good questions we can ask. And I do believe that we have pretty good answers currently, but the research being done, at least between myself and Craig, seems promising, Brian.
Awesome. Also another thing is with her saying like the family the genes and stuff, and you were talking about how who's classifying these things. What she's doing is probably like a fallacy. I think it's called I don't know, was it nomina fallacy? It's basically where you kind of assume that the name of something means that that this is how it should be defined. Like it's a butterfly, so it's a stick of butter with wings or something like that.
Yeah,
like that. But yeah, it could be named
a joystick.
Yeah,
A joystick.
it could be named for a lot of different reasons because we're classifying these things. And so what she's doing, she's assuming that because that we are identifying it as part of a family of genes and we're calling it DDX11L2 or L gene, DDX11L gene, that's the family of genes, and they were classifying it that, and then they say, well, then it must be telomere specific. Well, like you said, for one thing, it's subtelomeric anyhow. Right. But another thing is that this is being classified that way by them. So, is there any non-arbitrary reason why they would put it in a particular family of genes?
Yeah,
I want to make sure I leave no stone unturned on that in case someone brings that up.
Yeah, well, you raise some great questions, and it's the same questions I have. For example, in my book, Endogenous Retroviruses, I point out that your ERV sequences or elements, they didn't name themselves endogenous retroviruses. And you know what? That's probably not the best name for them, because anytime people hear the word virus, they think of something bad. Ooh, virus, right? Infectious, harmful in some way. And I point into my book, I think we should call them created units of DNA function. Some creationists have said we should classify them in a group of functional elements called variation-inducing genetic elements. Pseudogenes. We didn't name these DNA elements pseudogenes. The evolutionists did. And so the question is, should this gene actually be called the DDX11L2 gene? How do we know it belongs to that family? We definitely don't know that it used to be a protein coding gene that lost its ability to produce a functioning protein. It's not a pseudogene. It's not a genetic mistake. It's highly functional. And so I think these questions are important, and I think the evolutionists need to answer them.
So they might have classified it that way because they assumed that this is a telomere region in the first place, telomere fusion. So But okay, it's based on their own personal classification. But then Erika turns around and uses that to say, well, see there, so that proves that it is Tim. So it seems like the argument is circular.
It does seem circular. Also, this gene within the gene family, maybe they're necessary to be situated next to telomeres, close to telomeres, and also internal telomeric sequences like we find at the fusion site, right? They could be there for chromosomal stability, chromosomal architecture, structure, the health of the chromosome. They could help the function of the internal telomeric sequences. So, there's plenty of reasons for sure. But yeah, these are questions that we're actively looking for. Again, the research we've done on it, because there isn't really any creationists researching this specific issue, but it seems like there's been at least one other so-called gene in that family that's not next to a telomere and seems to be next to a centromeric DNA. That's preliminary though. So, don't quote me as that being infallible. I'm just saying that there's a lot more questions that the evolutionists, I think, need to answer for sure.
Right. All right. If you don't have anything else to add, we're going to continue.
Yes, I think that's good.
Not answer the question at all why there is a telomere specific pseudogene in the middle of human chromosomes. Oh, dang it. Okay.
There actually had human specific repetitive elements in it that you don't even find in apes.
There are specific repetitive elements that are species specific, right? Like he's trying to draw this dichotomy. You find this stuff in humans and you find this stuff in apes. There is no way to do that, right? If you want to say you find human specific stuff, then whatever that thing is that you're looking at, take orphan genes, for example. They're human specific orphan genes. There are also chimp specific orphan genes and gorilla specific orphan genes and orangutan specific orphan genes. They're only found in them. But there are orphan genes that are ape specific that don't include humans. You see what I mean?
And so,
Okay. Do you know what she's arguing there and whether it's valid or not?
Yes. I'll share my screen and show why the model that I hold to, and the model that Christopher Roop holds to, and a lot of young Earth creationists, her argument doesn't provide us with a challenge or a problem, because you'll notice the phylogeny here. This is the separate ancestry model. This is what I hold to, and this is what I've been confirming using mitochondrial DNA, Y chromosome, orphan genes, DNA function, other taxonomically restricted genes, linkage blocks. Okay, so
right.
Yes, we would agree that a lot of these sequences and DNA elements are species specific. But you know what? It falls in line with our model here. So you got Homo sapiens, humans, that all belong to one kind. And then I would argue that you basically have, within your primates, you have gorillas as their own kind, chimpanzees and bonobos as their own kind, and then orangutans as their own kind. And so I wouldn't argue that your gorillas are related to your chimps. And so if there's species specific sequences or species specific orphan genes, it more or less lines up with the phylogeny I have here. Now, if I was a creationist who said, you know what, gorillas and chimps and bonobos all belong to the same kind, then there might be a challenge. That might be something I need to work out, right? But I think the data suggests that gorillas and chimps are their own specific kind. And what's funny is the Y chromosome data, Brian, trying to find it real quick for you, actually supports this. Notice this. This is from the secular literature. This is from the conventional literature. This is from a paper, The Y Chromosomes of the Great Apes, 2017. Notice the differences in Y chromosome. You got your human Y chromosome, which is totally different to the chimpanzee Y chromosome. Half the size, totally different in gene content. This makes sense. Humans and chimps are separate created kinds. But you'll notice the gorilla Y chromosome is actually more similar to the human Y than either are to the chimpanzee Y. And so again, this confirms what I'm saying. Humans are a separate kind. Chimpanzees and bonobos are a separate kind. Gorillas are their own separate kind. And then your orangutans are their own separate kind. And so you have multiple separate kinds here within primates. And so sure, some of these genes, some of these sequences are species specific, but overall the pattern seems to fit this phylogeny. Does that make sense and help, Brian?
Wow, that was incredibly cool. He brought presentations. There was all kinds of sorts of arguments against and responding to Erika concerning alleged chromosome 2 fusion. To make sure that we're clear, we're not saying that it's impossible, but it does seem very highly improbable, and the evidence seems to be stacked up against it as well. And the evidence that evolutionists use to favor it seems to be very weak at best. And of course, we're going to be continuing the discussion. Not next week, but within a couple weeks, we're going to be doing the part 3 of the Tompkins Controversy, in which case we're going to deal with the so-called junk DNA that Donny really focused on, and that's where he really shoots a lot of really powerful arguments against the evolutionary view on junk DNA, and as we're responding to some things that Erika said on it as well. And I think that's going to be very strong in favor of what's going to happen in the Tompkins controversy, because a lot of the stuff that they're cherry picking, some of the things they're cherry picking is like what they call the junk DNA and what they call pseudogenes and things like that. And so it'd be cool to kind of go through all that with Donny. We had that second part of our interview, but that's going to be the next time on the Tompkins Controversy. We're going to do that as a separate video. But he did some really, really good arguments in favor of that. Next week, we're going to be doing a response video of some sorts. I'm calling it the Tompkins Controversy Intermediates, where it just kind of takes like a timeout from the controversy of the Tompkins Controversy to discuss some things that are going on in the comment section, some things that are going on in video responses, or that they're previous to, they're either about previous videos or about or their video responses about the previous videos. And so I'm going to have that, and I want to wait until next week to do that, because Erika's supposed to be launching a response to Carter's video, and I think she's going to say some stuff on the Richard Buggs issue too, as well. So I can't wait till she throws that up there, because she gave me only a few hints, but my gut tells me I know exactly which direction she's going into. If she does, I know exactly how to respond to her. But I kind of figured she would try to twist it around a little bit, because she had some very public errors that was exposed. You know, her errors have been exposed in a very public way, and so now she's going to be. This is probably Erika at her most dangerous. And of course, by dangerous, I don't mean that we're shaking in our boots kind of dangerous. I've seen her kind of response videos. They're usually not very good. So I'm not expecting a whole lot from this. But what I am saying when I say dangerous, what I mean is that she's going to be like a trapped animal trying to get free, and she's going to try to fight and fight and fight and fight. And so she's going to be very desperate, because her errors have been exposed in a very public way, and now she's going to be very desperate. And this could make her dangerous in terms of the forcefulness of her arguments. It's not the rhetoric of her arguments. Her rhetoric arguments going to be very weak, I'm guessing, based on previous experience dealing with Erika's arguments. But she's going to be very forceful about it, and she has a lot of influence from a lot of evolutionists. So a lot of evolutionists are going to probably take her claims and run with it, which I think is kind of hypocritical, because she tried to claim in the emails, probably because Ruhib told him, because Ruhib tried to accuse me of the same thing, that I'm just parroting Tompkins and Carter. That's not true. That's not true. I piggyback a lot of stuff they have because it's really good content, but I'm not just parroting it. But that's false. But in any given case, I think it's so hypocritical that she says that about me, but yet her followers actually parrot her. So anyway, but But whenever you repeat something that somebody made lots of errors in it, chances are you're going to make some errors as well. And when her followers repeat her statements, and she's got a lot of errors in her claims, they're going to repeat her errors, is what's going to happen. But that's what I mean by dangerous. I don't actually mean that, oh no, we're shaking in our boots kind of dangerous. I mean, she's going to be much more forceful about her arguments. She's going to be much more desperate to hang on. Based on what she told me in the email, she has no intention of stopping until everyone pretty much agrees with her on Tompkins, and I assume Tompkins is method is out to lunch, and then we can't trust anything that Tompkins says and things like that. And she's really got it out for Tompkins. I don't know why. I really don't know what her personal deal is with Tompkins, but she has some kind of personal vendetta against Tompkins, and I do not know why. I've never seen her like this with anybody else, even Dr. Jason Lowell. I mean, she's got a few videos against Dr. Jason Lowell, and she's even got she hates Ken Hovind with a passion. But I've never seen her like this with anyone else. So this is unusual. Maybe it's because Tompkins has a lot of support behind him. I don't know. But she really's got it out for Tompkins. But in any case, she's desperate. She's obsessing over it. So this is why I said she's going to be at her most dangerous. I don't mean that we're going to be shaking her boots or that she's going to have any strong arguments. I've seen the kind of arguments she has. I think that her arguments typically are weak, and so I don't expect much from them anymore. So I don't expect much from this, but I am kind of anxious to see how she responds to some of these errors that she's been exposed to. I thought she originally that she would admit to some of them, but I'm not so sure she will admit to any of them now. But we'll see. I would have to say that about Erika. This is going to be Erika at her most dangerous when she's backed up into a corner like this. And so she's going to come out trying to fight. And it's going to get messy. There's probably no other way around it. It's going to get messy. But I'm going to sit there and see what she says about it. She's also probably going to try falsely accuse me of doxing her in my original version of the video before I edited it. That wasn't true. I didn't dox her. There's going to be more about that next week, but I didn't actually dox her. I didn't search for this information or anything like that. This was actually just given to me by Bugs in a conversation with him, so I was just reading off the emails. But she assumed that I was trying to dox her, and I wasn't. Doxing has a very specific context and a very specific definition, and we'll be going over that next week because she did accuse me of allegedly doxing her. I didn't actually dox her. And the funny thing is, as soon as I found out that she didn't want her last name a matter of public viewing, I immediately went to it and corrected it. I fixed the situation. I was very prompt about it. I put the video on private so nobody else can view it while I was trying to get the editing process done. This was suggested by Dr. Dan from Creation Myths, and so I thought that was a pretty good advice, and so I took it. And I even apologized for a mishap, but that doesn't mean nothing. None of that stuff means anything to Erika. So she's going to probably raise it up. She's going to make a big deal out of it. She's going to accuse me of doxing her. I didn't dox her, but she's going to accuse me of that. And the strange thing is, I did not actually dox her. I can show you the definition of doxing. That wasn't doxing. But in any case. But that's going to be next week. And so, and it's going to be coupled with also responding to whatever she says about Richard Bugs, because I want to hear what she says and see what they are saying about that in the comment section. So I'm going to give that another week before I respond to those things. And I also want to, in that same video, I also want to respond to a few things that have been stated by Ruhif in the comment section as well. Some arguments that he made, but we're going to save that for next week. All right, and but thank you for joining me for the Tomkins Controversy. I really appreciate it. I can't wait for part 3 to come out with the second part of my interview with Donny Budinsky from Standing for Truth, because we're going to be discussing junk DNA, so-called junk DNA. And Donny is going to be bringing some very powerful arguments against their evolutionary views on this, and I can't wait. So thank you for watching. Don't forget to hit that thumbs up on the video. Don't forget to subscribe to my channel. Don't forget to hit that notification bell. Okay, be notified and alerted of all future videos. And thank you again for watching, and I'll see you on the next one.